You've probably heard people say things like, men are just more logical, or women are naturally better multitaskers. But what happens when science actually tests those claims? Over the last decade, large-scale brain imaging and meta-analyses have shown that while average size differences exist, the overlap is so great that simple labels like male brain and female brain don't hold up. Many regional differences even shrink or vanish once you correct for total brain volume. And just to be clear, we're talking here about biological sex assigned at birth. Most of these anatomy studies involve people whose gender identity aligns with that assignment, which means the findings don't automatically apply to transgender, non-binary, or intersex individuals. By the end of this video, you'll have a clearer, less binary way to understand cognitive strengths and challenges, and why this question still shows up in everyday conversations. Why this debate still matters in 2025. Walk into almost any workplace, classroom, or family gathering, and you'll still hear people casually mention a male brain or a female brain, as if those categories are fixed. The terms pop up in jokes, dating advice, or quick explanations for why people think differently. Behind these everyday remarks is the assumption that brains fall into neat biological boxes. Yet time and time again, when neuroscience puts those claims to the test, the results throw doubt on the simplicity of those divisions. By 2025, what we know points instead toward a far more complicated and overlapping reality. Most brain imaging datasets have been collected from people whose gender identity aligns with their birth-assigned sex, so we must be cautious applying those results to trans or intersex people. Within those limited samples, one of the most consistent findings has been that, on average, male brains are about 10 to 11% larger than female brains. But this difference mostly tracks overall body size, and it doesn't mean one is better or more advanced. Once size is accounted for, the overlap between individuals is enormous, so large that you can't reliably predict how someone thinks or learns just by looking at whether they are male or female. This single statistic highlights the tension between cultural expectations and scientific evidence. People want clear labels, but the data resists being boxed in. That desire for categories is ancient. For centuries, societies described men as logical and women as emotional, often using those arguments to justify who should lead and who should nurture. Those beliefs shaped education, medicine, and policy. But like many inherited assumptions, they became so familiar that repetition made them feel true, even without supportive evidence. Modern neuroscience has now confronted that legacy by showing that individual brains often mix traits once believed to divide cleanly along sex lines. One person may display structural features considered typical of the opposite sex in older research, and none of that undermines healthy function. This matters because the effects are not only historical, they continue to shape systems we rely on today. In medicine, stereotypes can distort diagnoses. For example, depression has long been considered more common in women, but evidence shows men often present with different symptoms that traditional diagnostic models overlook. Similarly, ADHD has been labeled a male condition, leading to underdiagnosis in women whose symptoms may not fit the expected pattern. Anchoring diagnosis to rigid categories can misdirect treatment, leaving people without the care they need. Drug development has faced similar pitfalls. Clinical trials that assume brain differences are strictly binary may optimize treatment for one group, while failing to detect side effects in others. Dosages based on an imagined typical brain risk ignoring meaningful variation, and create imbalances in how safe or effective therapies are across populations. When the categories themselves are fuzzy, medical research based on those categories becomes less precise, sometimes even harmful. Education offers another clear example. If a teacher assumes boys are naturally stronger in problem solving and girls are naturally more empathetic, the classroom environment itself starts shaping student outcomes. Researchers point out that small behavioral differences can get amplified by adult expectations, creating self-fulfilling patterns. A girl discouraged from pursuing technical fields may internalize limits that began not with her brain, but with how her brain was described. Over time, these cumulative influences can become misread as hardwired truths when they may actually be results of unequal opportunity. Seen in this way, the central question for 2025 isn't whether brains vary between sexes. We know some patterns exist on average. The deeper issue is whether dividing human cognition into two opposing categories adds anything meaningful to science, medicine, or social life. Increasingly, the evidence suggests it does not. Neuroscience reveals tendencies, distributions, and overlaps that are far more complex than the binary framework implies. Averages may exist, but averages also hide enormous variation. The danger of binary labels is that they travel easily, detached from their academic context. A study reporting an average statistical difference between groups can transform into a viral headline declaring that men's and women's brains are different, full stop. On platforms like TikTok or in dinner table conversations, nuance disappears. What began as a cautious scientific observation is reinterpreted as proof of ingrained opposites, fueling stereotypes rather than clarifying them. That dynamic makes careful communication as important as the research itself. So why underline this debate in 2025? Because the costs of oversimplification are real. Misapplied categories warp medical practice, distort education, and reinforce social bias. Meanwhile, the more careful picture drawn from research shows that each brain is a mosaic, complex, variable, and shaped by both biology and experience. Treating brains as if they belonged neatly to male or female boxes actually reduces accuracy, rather than enhancing it. Which leaves us with a natural question. If broad size differences don't tell us much, and binary labels lead us astray, then what features actually matter? To move forward, researchers suggest testing 3 areas of focus that could give us better insight. Whether size versus structural differences make a meaningful impact, how hormones influence brain development and function, and how plasticity allows brains to adapt beyond what biology alone would suggest. And that brings us to the next puzzle. When you place 2 brains side by side, and one is simply bigger, does that difference really mean anything? The size versus structure puzzle. When scientists measure brains, one of the first things they notice is that, on average, men's brains are larger than women's. Large-scale datasets, like the UK Biobank, consistently report about an 11% size difference. That figure sounds dramatic until you account for total body size. Larger bodies require more neural resources to control muscles, organs, and sensory systems, so bigger brains are often proportional to the body they serve rather than a marker of higher capacity. After statistically adjusting for overall brain volume, what researchers call total intracranial volume, many apparent sex-based differences in regional anatomy shrink or disappear. What looked at first like a sweeping biological divide often turns out to be a simple scaling effect. It's also important to understand how misleading averages can be. Even though men's brains are larger on average, the distribution between individuals overlaps so heavily that the trait becomes almost useless for describing any one person. Many women have larger brains than many men, just as many shorter men have bigger brains than taller men. Size may be one of the most frequently repeated facts about brains, but its explanatory power is shallow. Simply pointing to bulk alone tells us very little about memory, reasoning, or creativity. Part of the persistence of the bigger is better myth comes from a false intuition. Larger brains should mean more neurons, and therefore more power. But biology doesn't work like computer processors. A whale or an elephant has a far bigger brain than a human, yet those species don't outperform humans in abstract thought or symbolic language. Within our species, too, sheer size does not map neatly onto greater intellectual performance. The efficiency of connections, not the raw count of neurons, seems to matter most. This is where modern imaging has changed the game. 2 decades ago, brain science had less detail. Researchers could note size, but not easily trace networks. Now, approaches like fMRI and diffusion tensor imaging show how information travels along fiber pathways, how regions synchronize, and how quickly signals move. Brains can be large but inefficient, or more compact yet wired for fast and flexible transfer between regions. These microstructural characteristics, folding patterns, density of neurons, connectivity maps, affect performance far more consistently than bulk volume alone. Cortical thickness has sometimes been highlighted as another anatomical point of difference, with some studies reporting slightly thicker cortex regions in women on average. But here again, the overlap is substantial, and results vary across studies. Researchers are careful not to make categorical claims, because the links between routine anatomical measurements and stable cognitive differences are weak. Outside of injury or pathology, brains of different sizes and cortical profiles generally support the same mental capabilities. In short, there is little strong evidence tying typical structural differences by sex to robust, consistent cognitive outcomes. The broader picture is that anatomy can vary in small group averages, But these distinctions don't translate into fixed psychological categories. A person with a smaller hippocampus can still perform well in memory tasks, and an individual with a larger amygdala does not automatically exhibit higher emotional reactivity. The relationship between region size and behavior is inconsistent across studies, which cautions against declaring that anatomy from one group predicts capacity or temperament for that group as a whole. What researchers emphasize instead is connectivity. Brain networks resemble transport systems in a city, where the placement and efficiency of roads matter more than how much land the city occupies. A densely connected brain can shuttle information with minimal delay, while a larger but loosely wired brain might encounter bottlenecks. These networks don't just differ between people. They adapt across time in response to learning, stress, and experience. Cognitive strengths emerge less from overall mass, and more from the flexible architecture and synchrony that structure supports. This reframing also changes how we treat the public narrative. People often latch onto statements like, "men's brains are bigger," without the necessary qualification that the effect disappears with body size adjustment, and that overlap overshadows averages. In reality, the differences are statistically small, practically limited, and not reliable guides for how someone thinks, remembers, or behaves. The persistence of size-based explanations reflects cultural bias more than scientific utility. Taking apart this myth also undercuts one of the historical arguments used to rank abilities. For centuries, size was conflated with superiority, whether in skull measurements or broader claims about gender capacity. By 2025, science has dismantled that reasoning. What matters is not how much tissue a brain carries, but how effectively that tissue is organized and used. Any attempt to attach intellectual value to sheer volume ends up ignoring the hidden architecture that actually drives mental life. So although brain size often makes the headlines, the weight of evidence shows that structure, wiring, and efficiency matter far more. Size differences exist and can be measured, but they explain little about who we are or what we can learn. For that, neuroscience has shifted its questions away from bulk and toward processes that shape behavior moment to moment. Size may have been the first and simplest way to compare brains, but the real insights come from how networks interact and adapt. And that leads into the next layer of the discussion. If structural traits and connectivity matter more than bulk, what about the chemical signals that steer those networks from day to day? Hormones and the brain's daily shifts. Hormones often become the punchline in casual conversation. If someone gets irritable, distracted, or overly emotional, people quickly chalk it up to hormones. Pop culture has branded testosterone as the aggression hormone and estrogen as the emotional hormone. But the science paints a more intricate picture. Far from being on-off switches that dictate behavior, sex steroid hormones like estrogen, progesterone, and testosterone act as modulators inside the brain. They influence the sensitivity of neural circuits, shape how neurotransmitter systems respond, and fine-tune plasticity at synapses. Their role is less about directly scripting behavior and more about shifting the conditions under which neurons interact. Estrogens, for example, have been shown in animal models and some human studies to promote synaptic plasticity, particularly in brain regions involved in working memory and verbal processing. Specifically, 17-beta-estradiol can enhance the formation of dendritic spines in the hippocampus, supporting networks involved in learning and recall. These aren't dramatic rewrites of how a person thinks, but subtle boosts that create windows of efficiency the brain can take advantage of. Testosterone, meanwhile, interacts with dopamine systems and networks involved in motivation and reward. Rather than forcing aggression or competitiveness, it adjusts the likelihood that certain pathways will activate, depending on context and prior experience. The rhythms of these hormones vary by sex and by life stage. Females experience cyclical fluctuations tied to reproductive biology, including menstrual cycles, pregnancy, and menopause. These cycles introduce changes in hormone concentrations that may influence aspects of cognition and mood over days or weeks. Males, on the other hand, show clearer diurnal variations, with testosterone levels typically peaking in the morning and declining through the day, as well as gradual declines across the lifespan. Both sexes live inside these shifting hormonal landscapes, but the timing, amplitude, and patterns differ. Importantly, neither pattern guarantees fixed behavioral outcomes. Any cognitive shifts are probabilistic and often modest. For instance, studies examining memory performance across the menstrual cycle sometimes report small boosts to verbal memory at points when estrogen is higher. But reviews consistently emphasize that findings are inconsistent. Effects vary from one study to the next, and, when present, are often subtle. In men, higher morning testosterone has been linked to slightly improved spatial or working memory in certain tasks. But here too, the effect sizes are small and not reliably observed across all individuals. These examples illustrate the point. Hormones set conditions that can make some cognitive processes a bit more efficient at particular times, but experience, sleep, nutrition, and stress often outweigh these biological rhythms in practice. This nuance also dismantles older stereotypes. Testosterone can increase the likelihood of competitive or status-seeking behavior, but it does not make a man aggressive in all settings. Social cues and goals matter. Rising testosterone can just as easily fuel persistence in academic work as it can fuel intensity in sport. Similarly, while estrogen interacts with networks involved in emotional regulation and social processing, that does not mean women are destined to be ruled by emotion when estrogen is high. In some studies, estrogen enhances sensitivity to emotional nuance, potentially improving empathy and communication. Attributes once framed as weaknesses are in reality context-dependent strengths or neutral shifts. Clinical science has also shown why these distinctions matter. Early hopes that hormone therapies could neatly preserve cognitive health, especially for women approaching menopause, have not been borne out in large-scale trials. Both the KEEPS and ELITE studies, two major investigations into estrogen therapy, found little consistent support for the idea that simply giving estrogen at the right time produces reliable cognitive protection. The mixed results highlight again that hormones are modulators, not magic levers. Cognitive outcomes depend on many interacting factors, and no hormone acts as a direct cure or guaranteed enhancer. An accurate way to picture these effects is not as a switch being flipped, but as adjustments to thresholds. Hormones modulate how excitable certain circuits are, which neurotransmitters are favored, and how easily plastic changes occur. Rising estradiol, for instance, might lower the threshold for encoding new memory traces, while declining levels might reduce that efficiency. A surge in testosterone could sharpen motivation, but whether that motivation is expressed in cooperation or competition will depend on personality, training, and environment. The same biological cue can support multiple behavioral outcomes depending on context. Environment and culture play a decisive role in shaping how these modulatory signals translate into action. Two people with comparable hormonal profiles may respond in different ways because of upbringing, current stress levels, or the cues around them. A rise in testosterone might prompt one person to focus single-mindedly on achieving a personal goal, while another interprets the same biological push as a cue toward interpersonal dominance. Neurobiology provides the backdrop, but environment supplies the script. This more balanced understanding is changing medicine. For decades, pharmaceutical research often ignored or smoothed over hormone-related variability. Trials were scheduled without considering menstrual phases, meaning potential differences in effectiveness or side effects were hidden by inconsistent timing. Now, researchers are beginning to align treatment strategies with these rhythms. The question isn't whether women and men are wired differently. It's how daily or monthly oscillations in hormone concentrations might affect drug metabolism, cognitive performance, or susceptibility to stress at particular times. Mapping treatments onto these rhythms may lead to more tailored, effective care. The same principle applies outside of medicine. Understanding one's hormonal patterns can enable practical adjustments without surrendering autonomy. Some individuals find it useful to track patterns in focus or mood across the month, using that information to match demanding tasks to periods when they feel mentally sharper. Others use awareness of typical dips to practice deliberate coping strategies. Recognizing hormones as moderators of cognitive texture provides a tool for self-awareness, not a strict set of instructions. What matters most is rejecting the outdated idea that biology writes character in bold, unerasable ink. Hormones set the stage for subtle shifts in cognition and affect, but the brain adapts around these shifts with a high degree of flexibility. Cognitive systems recruit alternative pathways, learning refines responses, and conscious strategies regulate behavior far beyond what ebbing and flowing molecules can dictate. Framing hormones as simple causes of personality or mood swings misses both the complexity of neurobiology and the adaptability of human experience. And yet, despite mounting evidence, simplified stories about testosterone and estrogen still influence popular thinking. That persistence isn't random. It reflects a history in which crude generalizations about sex differences were carried into research design itself, affecting what kinds of experiments were conducted and which groups were even included. Those choices left significant blind spots in the scientific record, gaps that still shape medicine and neuroscience today. The hidden gender gap in neuroscience research. For much of modern neuroscience, the picture of the human brain was drawn from only part of the population. Study after study leaned on male subjects, both in lab animals and in people. To most outsiders, this might sound like a technical detail, but it has shaped the evidence base in ways that ripple through how we define disorders, diagnose symptoms, and prescribe treatments. The imbalance was systematic, not incidental, and its consequences remain visible today. In basic research, the gap was especially stark. Early counts of neuroscience studies estimated that male animals were used about 5 times more often than female ones. Researchers argued that by avoiding females, they could control for variability linked to hormonal cycles. The logic was simple. Fluctuating hormones meant more statistical noise, making experiments less clean. What it created instead was a deliberately narrowed dataset that erased half the biology in question. The justification, however tidy, ignored the fact that hormones are part of real life. Cycles of estrogen and progesterone in females are not distractions from brain function, but elements of it. They modulate synaptic plasticity, neurotransmitter systems, and how circuits respond to stress. By filtering out that variation, scientists simplified experiments, but also excluded data that might have revealed different vulnerabilities or protective factors across sexes. For decades, female biology was set aside as a problem to avoid rather than a key part of the neural system worth studying. That decision has had lasting consequences. Medications tested primarily on men often worked differently once prescribed more broadly. Drugs optimized in male-dominated trials sometimes produce stronger side effects or weaker benefits in women. This played out in everything from antidepressants to sleep aids. Dosages that looked effective in trials often turned out less safe or less effective in women. Only after widespread use did some of these gaps become apparent. They weren't mysterious side effects. They were predictable outcomes of limited evidence. Neuroscience, in particular, lagged behind other specialties. While fields like cardiology began to deliberately recruit more balanced samples in the 1990s and 2000s, brain studies clung to older habits. Many labs hesitated to disrupt decades of male-centered data collection, worried about comparability or about the cost of adding analytic layers to handle hormonal changes. Some simply argued the complexity was unmanageable. In practice, this meant conclusions about brain structure and function were comfortable to apply to men, but less accurate for women. The consequences extend beyond drug safety. When diagnostic categories were shaped on male-heavy samples, the typical case often looked male. Conditions such as ADHD or autism were described in ways that aligned with how they appeared in boys and men. Women and girls who showed different patterns, more subtle presentations, different comorbidities, were frequently overlooked or misdiagnosed. Even depression, which occurs at higher rates in women, was initially modeled around male cohorts in trials, blunting recognition of how differently symptoms can manifest. Bias in subjects became baked into the definitions of disorders themselves. Correcting for this decades-long exclusion has only recently begun. In 2016, the U.S. National Institutes of Health introduced its sex as a biological variable policy, requiring researchers to factor sex into experimental design. This move forced labs to reconsider old habits and pushed grant applications to justify imbalances if they persisted. Implementation has expanded the representation of females in both clinical and animal studies, but it has also created new challenges. Handling hormone-driven variability adds statistical complexity, and not every study fully analyzes the data they now collect. Inclusion has improved, but many datasets remain male-biased or treat sex as an afterthought. The culture is shifting, but unevenly. Still, the effort has begun revealing what was missing. Certain patterns now recognized as clinically significant had lurked unseen. For example, some enzymes in the liver metabolize drugs differently depending on hormonal environment, altering how much active compound reaches the brain. Differences once dismissed as anomalies turned out to be common in women, but invisible in male-led samples. Imaging studies are also beginning to detect anatomical subtleties that explain why neurological or psychiatric conditions affect men and women at different rates or in different ways. Each addition makes the overall model more accurate, and each one points to how much nuance was lost by excluding female bodies in the first place. The lag also highlights how hard it is to revisit entrenched assumptions. Entire curriculums, textbooks, and clinical guidelines were built on research that systematically ignored variation between sexes. Rechecking those conclusions takes time and funding, and in the meantime, outdated models still circulate in classrooms and clinics. Students learning from old charts may unknowingly pass forward the very patterns of bias that newer research tries to correct. What looked like consensus in the literature often only reflected male-centered trends. The ethical dimension is unavoidable too. If science positions itself as an objective pursuit, ignoring half the population undermines that claim.